right, welcome everyone to our very last biochemically review of the semester. I know you guys have your final exam obviously next week. So today we're going to be talking about the electron transport chain, which is fairly extensive, unfortunately. But that's it. There's no more next week. There's no more, what's next. It's the electron transport chain. And then we're done with the entire process of oxidative phosphorylation. So yeah, I'm just going to go ahead and jump right in. So last week we talked about the tricarboxylic acid cycle, the citric acid cycle, the Krebs cycle, whatever we want to call it. And one of the biggest things that we discussed was that it creates some ATP, not a lot, but some, and then it creates a ton of NADH and FADH too. This is where we finally get that huge payoff, right? So glycolysis gives us a net of 2 ATP. Excuse me. The TCA cycle gives us a net of another 2 ATP. We're making very little, I should say per one glucose molecule. Let me make sure I'm clear about that. We're making very little energy from these. However, the electron transport chain, or as I'm going to start calling it ETC because it is a lot shorter, is the big payoff. This is where we get a ton of ATP, over 30. Okay, so I'm just going to go ahead and get started. So first is location. I told you guys about the importance of compartmentalization, location, location, location. And in this case, or I should say in eukaryotes, the electron transport chain occurs in the mitochondria, and specifically in the inner membrane of the mitochondria. So I believe we'll be seeing a picture right here. Yep, and I'm actually going to zoom in a little bit because I really want to emphasize this. So mitochondria also have 2 membranes. Okay, so there's this outer membrane. So this would be outside. So this is the cytoplasm. And then between the outer membrane and the inner membrane, there's what's called this intermembrane space. So this would be the intermembrane space. And then this right here is the inner membrane. And then this is the mitochondrial matrix. So this part is the matrix. Okay. Cristae are these kind of what you call, they're stacks of, or I shouldn't say organelles, of cells that create the mitochondrial, the inner mitochondrial membrane, excuse me. You'll learn more about those in biochemistry too if you take it. They're not super important, but for now, just keep these terms in mind. Outer membrane, inner membrane, intermembrane space, and matrix. That's actually, I like to remember it kind of like sequentially, if that makes sense. So too far, there we go. So I think of it as cytoplasm, and then I draw a line. This line is the outer membrane. Then we have the intermembrane space. Then the inner membrane. And then finally the mitochondrial matrix, which I'm just going to say matrix for. So this is the order of excuse me. This is the order of the kind of the structure of the mitochondria. So a couple of the things that we need to talk about are, I told you all about proton gradients. We talked a lot about how compartmentalization affects things. So when we say, the inner membrane has a potential difference, and we're going to be talking a lot about potential and electric cells in a little bit. What we mean by that is one side of the inner membrane, so the intermembrane space, we'll say, is very, very positively charged relative to the mitochondrial matrix, and therefore there's a potential difference. Okay. Again, that's something we're going to get to. I just want to make sure that the notation is clear. Okay. So standard reduction potential, which we use the Greek letter epsilon naught for, and the naught refers to that standard. So how delta G is free energy, whereas delta G naught is standard free energy. Same thing here, except it's a subscript instead of a superscript. Energy. Okay. So basically what we see is the basics of redox reactions. So I'm going to do like a quick 30-second recap of what a redox reaction is. A redox reaction, the word redox comes from reduction and oxidation. And these two processes work in concert. So you cannot have just an oxidation reaction or just a reduction reaction. You can separate the two using an electric cell or like a voltaic cell, a battery, or things like it. But in order to have a reduction, you have to have an oxidation and vice versa. Okay? And the reason that this has to happen is because redox reactions are electron transfer reactions. Now, why this is important is reduction is a decrease in oxidation state. In other words, it's accepting more electrons. Where is it getting those electrons from? From the oxidized donor. So as you see here, we call them donors and acceptors. That's one term for it. I call it the species that gets reduced, the species that gets oxidized. All of these are acceptable terms. But the donor that gets oxidized releases its electrons. And when it releases those electrons, they go to the acceptor, which then gets reduced. And so as a result, these reactions have to happen together. You have to have electrons in order to reduce something. You have to release electrons in order to oxidize something. But electrons aren't just going to float around in space, forever. And so because of that, you have to oxidize one in order to reduce the other. Okay. And then finally, down at the bottom, you guys see the, I shouldn't say the bottom, toward the middle, you guys see the equation that relates free energy to epsilon naught. So again, just very briefly, I want us to consider these values and numbers and stuff. So N is the number of electrons. The number of electrons can change because, for example, aluminum can have a +3 charge and go to being uncharged. In that case, it's accepting 3 electrons. On the other hand, you can have something like sodium. Sodium takes on a +1 charge, and to get to a neutral state, it accepts a single electron. Excuse me. And so N, number of electrons, is the number of electrons that gets transferred from the donor to the acceptor. Okay, and so this is variable, which is why I have to account for it. F is called the Faraday constant, developed by Faraday, or I shouldn't say developed, discovered by Faraday. And it's a positive number is the point. That's just the key here. So N is always a positive number. You can't transfer a negative number of electrons. Faraday's constant is also positive. So therefore, when we think about free energy, I always think about free energy as negative is good, right? We want it to be spontaneous. We want this number to be negative. There's a negative out front that I want to highlight to emphasize it real quick. So this negative is out front, right? Number of electrons in Faraday's will always be positive. Therefore, if E naught, and we'll talk about why there's a delta here in a moment, if delta E naught is a positive number, we have a spontaneous reaction because then delta G naught is negative. If delta E naught is a negative number, we have a non-spontaneous reaction because that would make delta G naught a positive number. Okay, so now let's read down here. Delta epsilon naught is the difference in reduction potentials between donor and acceptor. Okay, so thinking back to Gen Chem, must have been Gen Chem 2 at this point, when we look at these standard reduction potentials, this has a certain value to it. So maybe this is negative 0.8 volts. And maybe this one is negative 0.2 volts, right? And so if we can pair these in such a way, we're going to have to flip one of them since both of these are reductions. So if we flipped this reaction, the new epsilon naught would be 0.2 volts positive, right? And so we have to figure out a way to make this positive in order to make delta G naught work. Okay, so now I am personally not a huge fan of this notation, or this formula, I should say, for delta epsilon naught. And I apologize, I'm probably going to call it E. I think I already have called it E. So just for the record, E and epsilon are not interchangeable, but I might accidentally use them interchangeably. Okay, so this is an acceptable formula to use. I just don't like it very much. Because we're given values as standard reduction potentials, we always have to flip one. So let's use that example I had above. So E minus gives us Na, and I'm not going to include signs, but this would be aqueous to solid. And the epsilon of this reaction, we'll say, is that negative 0.2 volts number. Okay. Aluminum, 3 plus 3 E minus, going to aluminum solid, is going to have an E of negative 0.8 volts. Okay, and we're not going to worry about it being different numbers of electrons for now. I'll show you why it doesn't matter later, but just bear with me for this. Okay, so what this equation is telling you is if I were to say negative 0.8 minus negative 0.2 equals negative 0.6 volts, that's telling me that electrons will, I should say that the negative 0.8, so the aluminum, is not the acceptor, and the sodium is not the donor. If I flipped this on the other hand, negative 0.2 minus negative 0.8, I get positive 0.6 volts, and therefore the sodium has to be the acceptor and the aluminum has to be the donor. Now again, this equation works, and again, I don't like it very much. The reason that I don't like it is because I think this is a better way to think of it, of oxidation, and reduction. Okay, so the formula that I've written out is the epsilon naught of the oxidation plus the epsilon naught of the reduction. Okay, when we look at these two formulas, you can tell me which one we have to flip in order to make a positive number, right? If I make this negative, or if I say if I negate this number and make it positive as a result, that will make this overall positive. And so therefore, I know I have to flip this one. I have to flip this equation. Since this is the reduction, that means that I'm going to end up oxidizing aluminum. So what is the potential, the E of the oxidation now? It's not negative 0.8, it's negative 0.8. In other words, it's plus 0.8. Then the epsilon of the reduction is minus 0.2. And all we have to do now is add them together. So the reason that I like this formula a little bit better than the other one is because, first of all, it comes from intuition. There's no need to figure out which one is the acceptor, which one is the donor. There's no need to memorize the subtraction. It's just add the two, flip the one you need to flip, and then flip that reaction as a result. I think that that's more intuitive. Okay, you can use either. You can use, I'm sure there's other variations that I don't have off the top of my head. If it works, feel free to use it. I'm most comfortable with the one on the right, so I'm very likely going to use this one for the rest of this review. But yeah, again, any of them are totally acceptable. Okay, or exactly what I'm saying here, this point right here is also really valuable. Okay, the redox couple with the more negative reduction potential will donate electrons to the couple with the more positive reduction potential. So let's translate that from chemistry talk into like real actionable information. So when it says redox couple, it means Al3 plus to Al. It means sodium plus to sodium. Okay, and so the more negative reduction potential in this case is the negative 0.8. Okay, it's going to donate its electrons to the couple with the more positive, which in this case is the sodium. So therefore, this is going to be the donor. So if you memorize that, figuring out which one is the donor and which one is the acceptor becomes really easy. Or if you memorize just that first half, just based on math, if these are both negative numbers, I need to flip the bigger number in terms of absolute value in order to make a positive number. Please excuse me. So if I need to flip, if I flipped 0.2 to a positive number, negative 0.8 plus 0.2 is still negative 0.6. It doesn't help me anything. If I flip negative 0.8 to positive 0.8, I then get a positive number because I'm adding a number smaller than it. Okay, and so just thinking of the first part, the more negative reduction potential flips. The more negative reduction becomes a more positive oxidation. That's how I think of it, and I think that that's the more intuitive way to think of it. Okay, all right. So your professors include some videos and some other content that I don't think is quite as important. I just want to talk about this for now. So I told you all, reduction and oxidation have to happen in concert. You cannot have one without the other, okay? Scientists have gotten really clever. And so we're not doing one without the other, we're just separating them into different containers. And this is how batteries work, by the way. Okay? So what we do here is we have two electrodes, okay? And those electrodes I'm circling one of them in blue, but I don't think you guys can see it very well. So we're just the electrodes are the metal bits, they're goldish, bronzish color. Connected to them is some kind of some kind of connection, just a metal or just anything that electrons can flow through. And a salt bridge. The salt bridge allows ions to flow through it, either this way or this way. The potentiometer isn't super necessary, but it does help, and I'll explain that in a bit. Then inside the solutions are the things that you may want to reduce or oxidize. Okay, so let's start off with what we're looking at here. We're looking at the something of ethanol to acetaldehyde. Maybe it's a reduction, maybe it's an oxidation. We're not 100% sure yet. Okay, so this is like I said, this is how battery works, but it's also how we determine the standard reduction potential. So I'm not going to spend too much time on this concept because you just need to know that you'll be provided a table of standard reduction potentials for anything you need to solve for. This is just a little bit wordy, but we're just going to go through it quickly. So important note is standard reduction potential refers to, once again, 1 molar solutions, okay? Just like standard state, 1 molar solutions. And yes, there is that biochemical standard state. Once again, we'll talk about that later, okay? For reference, the oxidation of H plus to H2 gas is defined as 0 volts, okay? So the important thing in our formulas is not the actual number. It doesn't matter that ethanol to acetaldehyde is 400 volts. That's arbitrary. It could be 300 and you would just subtract everything else in the table by 100 and everything would work the same. What matters is the difference. Remember that our formula is delta G naught is equal to negative N times F times delta epsilon naught. And the key here is this delta, okay? The only thing we care about is the difference in potential, okay? We don't care what the actual number is, we care what the difference between the numbers is. And that's why we can do this by setting this equal to 0, we create the table all the same. If we decided to add 12 to every single value for some reason, we could. If we decided to make this ethanol to acetaldehyde 0, we could. Any of that is fine, okay? All right, so that's it. Yeah, so the reference half cell is exactly what I was saying. It's assigned arbitrarily as 0 for convenience. Okay, so now, okay, there we go. This is what I wanted to emphasize. So once again, standard conditions, reactants are at 1 molar concentrations, H2 in the atmosphere is 1 atmosphere, and the pH is 0. We don't care about that. This is biochemistry. We want epsilon naught prime, okay? The convention in biochemistry, right? The standard biochemical potentials, if we want to think of it like that. So therefore, when we have a different pH, the reduction potential of H plus to H2 changes, okay? Despite H plus to H2 being our standard and always being defined as 0, it's defined as 0 for standard state, not for this, okay? That's the other thing important. Okay, so now let's deal with the numbers. So what we see here is this side is the reduction, this side is the oxidation. Okay, so ethanol is oxidized to acetaldehyde, H plus is reduced to H2. And in the process, 2 electrons are transferred. So when I put together my delta G naught formula, and we'll stick to delta G naught, although this could also be delta G naught prime, and I have negative NF delta epsilon naught. So the first thing to point out is that potentiometer shows negative 0.197. Okay? So that tells me that the standard reduction potential of this redox couple is negative 0.197. And then I told you, well, hang on, this is 0, this is negative 0.197. So the way that they do it down here to figure out the epsilon naught or delta epsilon naught is they say acceptor minus donor to get to this number. The way that I do it is flip the more negative number, make this now 0.197, and we're doing this as an oxidation instead. Therefore, it's going to be 0.197 plus 0, because this is the epsilon naught of the oxidation, this is the epsilon naught of the reduction. Okay, either way, we get to 0.197 as the total, right? And then if we wanted to, we could go ahead and calculate delta G naught. Because we have n equals 2, we have epsilon naught. The only thing missing is Faraday's constant, which is a number near 90,000. It's like about 95, 96,000. Okay, so this is what we care about. From everything we've just discussed for the past almost 20 minutes, this is the most important thing to focus on. This is the table of standard reduction potentials that we really care about, okay? So a couple of quick things to point out are, once again, this is always, always, always going to show reduction potentials, okay? So the thing with the highest reduction potential So oxygen, highest red potential, wants to be reduced. It's almost, I mean, if this table were all the what's it called, every element, every ion, everything that could possibly be oxidized or reduced, this thing could never be reduced. Or I'm sorry, it could never be oxidized. It's not, obviously, which is why it can be oxidized, but if, right? And then down here, so the lowest reduction potential is the worst at being reduced. At being reduced. Okay, so put together, what this means is the lower values on this table will flip to be oxidized. Okay, all right, I don't want to spend too much time on this because this is a general chemistry topic and we don't need to know a ton of it for the purposes of this class. But if you do have any questions about this, please feel free to stop by drop-in tutoring. We're open the rest of the week through Saturday. I'm unfortunately going to have to move on from this for the sake of time, though. Okay, so let's talk about what's important for us in this class, and this is the key here. Oxidative phosphorylation, remember, eventually transfers electrons. It reduces oxygen. Okay, and oxygen is very, very good at getting reduced. It's got a very high epsilon naught, right? And then we look at the NADH that we've been dealing with, negative 0.32, 0.816, epsilon naught equals negative 0.320. Okay, what this means is this wants to be oxidized, and in the process will reduce oxygen very quickly. Remember, oxidative phosphorylation, once again, that means oxygen is the final thing. I should say oxygen is the final reducing agent, I believe is the, no, I'm sorry. Oxygen is the final species reduced. I had that backwards, I apologize. Okay, all right. So this is a great example. So a standard redox can be used to predict the standard free energy. So what they've done here is they've put together, they've said this is the net equation. This is the first half of the redox reaction. This is the second half of the redox reaction, right? So there's one thing being reduced and one thing being oxidized. And then what we've said is since they're both reductions, we're going to take this lower one. This one will be oxidized, and so we will flip this reaction. So when we look at the Krebs cycle, we don't go alpha-ketoglutarate to isocitrate. We go isocitrate to alpha-ketoglutarate. Why? Because flipping this reaction makes it spontaneous. Okay, and as you'll see in the next page, they prove that. And then they say, okay, let's plug it in. And therefore, we get a delta G value, delta G not prime value, that's negative and therefore exergonic. Okay, so put together energy and what's it called? I should say free energy and equilibrium and electric potential kind of all give us the same idea. Okay, so just like with Gibbs free energy, you can have it in standard state or not in standard state. So Gibbs free energy, oops, I did not mean to draw that period. Let me erase it, or the circle. I don't want standard state. Just a reminder, Gibbs free energy is equal to delta G naught plus RT ln Q. This has some very, very similar aspects to it. The reason it has so many similar aspects is because it's basically the same thing. Why is it basically the same thing? Because all we're doing is we're saying, let's plug numbers in for these. So delta G we'll write as negative N times F times epsilon times, I'm sorry, delta epsilon. So this is not epsilon naught, right? Delta epsilon equals negative N times F times delta epsilon naught. So this is standard state plus RT ln Q. And then we've divided negative N times F from both of them, which is where you get this RT ln thing. And then the Q becomes oxidation over reduction. Okay, so these formulas are all interconnected. And so that's just kind of, wanted to briefly talk about that. That formula you should know. You can't be asked about these questions, totally fair game. Okay. So now in the ETC, oxygen is eventually used to re-oxidize NADH to NAD+, which makes it very, very, very spontaneous. Like, I know you guys see this number, negative 219 is ridiculously high. So when we look at ATP, ATP is negative 30-ish, this is negative 220, okay? And so because of that, we can create ATP using this. And we'll talk about all of that in a little bit. Before we talk about all of that, it's important to understand how we move these electrons and protons and all this stuff. So coenzyme Q or CoQ or Q or ubiquinone, there's a million different names for it, can accept up to 2 electrons to either become what's called an intermediate or a radical, or to become ubiquinol, which is the completely reduced form. Ubiquinone is 2 ketones, ubiquinol is 2 alcohols, okay? And so there's a couple of different terms. I'm usually going to stick to Q, QH, and QH2 just because they're shortest, but they all mean the same thing. Okay. There's also UQ, which is the same thing. Okay. All right. So this is what's going on in E. coli. I'm not really going to focus on E. coli because I really want to have enough time to talk about, the human body. So these are some of the basic electron transport mechanisms. But okay, this is what we care about. This is the human electron transport chain. So let's keep track of location. So intermembrane space to matrix, that makes this the inner mitochondrial membrane. Okay. All right. So there are 4 protein complexes embedded in the membrane. There is transport proteins, so UQ or ubiquinone, inside the membrane, and there's a transport protein outside the membrane, and that's the cytochrome C that you see up here. Okay. Now I want to point something out. I told you guys this, I told you all that succinate dehydrogenase would come up again. It just did. It's this thing right here called complex 2. There's a little, or I shouldn't say a little, there's a lot more to it that we do need to talk about, but the reaction that it's doing takes place right here on the inner mitochondrial membrane. Every other enzyme in the electron transport chain, I'm sorry, not in the electron transport chain, in the citric acid cycle is inside the matrix or inside, I think they're all inside the matrix now that I think about it. This is the only one embedded in the membrane, okay? All right. So you guys can see protons are being transferred, stuff like that. We're gonna get to each in a more detailed way, but effectively the end goal of the electron transport chain is the creation of a proton gradient, okay? And we'll talk about that, okay? I told you all last week that we're gonna bring up iron sulfur clusters again. Here they are. You guys can see them on the left in this figure. The big thing here to remember is iron sulfur clusters are great at moving electrons. Okay, that's as much as you need to know about them for most of this class. I have not seen exam questions that ask you how many iron sulfur centers does whatever have. That's not to say they're not fair game. So I suppose it's technically possible you'll get one of those questions, but I think there's more important things to talk about here. Okay, so real quick. The big thing here is this is the re-oxidation of NADH to NAD+. Yes, oxidation, okay? And so in doing so, we reduce FMN to FMNH2. FMN, remember, was the flavin mononucleotide, all right? And then we keep doing these kinds of redox transfers. So I call them redox transfers. I don't know if that's their formal name, because they're redox reactions that are being used to transfer these electrons a little bit down the line. So then we do another redox transfer to our iron sulfur center, then another to ubiquinone, which is then converted to ubiquinol. And then we do another to another iron sulfur cluster, and then so on until we finally get to the end of complex 1. So effectively what's happening is electrons are traveling all throughout until they reach the inside of the membrane, which is where the end of complex 1 is. Okay. Now, while this is happening, 4 electrons are being moved from the matrix to the intermembrane space. Okay. And there's kind of a lot of details. Okay, this is an important step. This diagram is wrong. I shouldn't say wrong. It's slightly inaccurate for convenience. So first, the iron sulfur clusters are not located as you guys can see them here. there are none in the membrane-associated portion. So all these iron sulfur clusters need to move out. Okay. Then second, it talks about ubiquinone being bound as a cofactor, but there's not really ubiquinone from what we've seen, right? This could still be proven incorrect, but for now we're just assuming no ubiquinone here. Okay. All right. Anyways, so this is kind of a slightly more accurate depiction of this. First, there are 9 iron sulfur clusters, and they're all in the what's it called? Not in the membrane associated part, excuse me. The only ubiquinone is not associated. It can freely leave. And yeah, okay. So we have those 4 protons that are moved in. We have the NADH to NAD+. Yeah, okay. This figure is just basically saying this redox reaction wants to happen. NADH will lose its electrons to become oxidized, and in doing so transfer them to FMN. So I like to think of this as, like I said, redox transfers, because we go reaction 1, reaction 1. This is involved in reaction 2, this is involved in reaction 2. This is in reaction 3, this is in reaction 3, and so on. We keep working our way up till we eventually make it to oxygen. And by the way, I have no clue which things I just pointed out here and here. They're completely arbitrary. I don't mean them to say these are the things to memorize. I'm just using it to kind of show that we're progressing our way up these standard reduction potentials. Okay? So yeah, so we don't know the exact path, but they go through the chain of iron sulfur clusters, and this is important. They each have a more positive reduction potential than the last. So this little reaction 1 to reaction 2 to reaction 3, it keeps working its way up even through the iron sulfur clusters. They're all slightly different, and that allows them to keep transferring these electrons. The electrons are ultimately taken by ubiquinone, and we'll talk about that after. Okay, so this is a weird figure. What this is saying is that there are 54 transmembrane helices and 7 subunits, and then in the hydrophilic region there are 8 subunits. So this is not super important, it's just a little bit of background. Okay, and then these are the iron sulfur clusters, and okay, so this is an interesting thing to think about. So we like to include more vocab because it can be helpful. So first, the periplasm. The periplasm is the intermembrane space. Okay, so I've been calling it intermembrane space this whole time. That's not exactly right. So the other thing is the cytoplasm / matrix. So cytoplasm is, yes, it's basically the cell. It's the inside of the cell, basically the liquid in it. It's also the inside of the mitochondria, because remember that the mitochondria has its own DNA and organelles and everything. It's almost like a mini cell, and so it's got its own cytoplasm. For convenience, we just call it the mitochondrial matrix because we don't want to confuse anybody by saying the inner cytoplasm and the outer cytoplasm or something like that, right? The periplasm is known as the intermembrane space. So in that diagram we drew before, it was outer membrane, and then out here was the cytoplasm of the cell. So I'm going to label that cell cytoplasm. And then there was the inner membrane, and then all the way in is the matrix, or the mitochondrial cytoplasm. This area right here was called the intermembrane space. Now we're just going to give it another name, which is periplasm. Okay, so there's an error here. What is that error? These two need to be swapped around. Okay, all right. So we've talked about ubiquinone, ubiquinol. It can accept 1 or 2 electrons, and it's a mobile carrier that's hydrophobic, meaning that it can exist inside the plasma membrane. Okay, all right. We're not going to talk about this too much. I don't want to spend too much time on complex 1 because the gist of it is basically 4 protons get transferred through, and then it gives coenzyme Q 2 electrons and 2 protons to become ubiquinol, which can then move stuff around. Okay. All right. Once again from last week, FMN, flavin mononucleotide, can either have a 1 electron or a 2 electron reduction. Not the most important thing right now. Okay. These are more figures that your professors are including to help you conceptualize. I unfortunately don't have time to go through all of them, so I'm just very quickly moving on. Okay, so like I said, we're hopefully good on basically the left half of this dotted line. 4 protons are transferred in, NADH is oxidized to NAD+, and in doing so, we transfer 2 electrons to coenzyme Q. What coenzyme Q does with them, we'll talk about later. The other complexes we'll talk about in a little bit, but I just want to mention that very briefly. Okay, so now complex 2, succinate dehydrogenase, serves a very similar purpose. And more specifically, it kind of serves 2 purposes. Purpose 1 is the actual dehydrogenation of succinate to fumarate, right? That's the obvious one. 2 is it deals with some protons and electrons, and it doesn't actually transfer protons through, but it helps us get electrons. So in the same way that this thing, we drew dotted line of electrons, this thing is also going to have a dotted line of electrons. Okay, so complex 2 creates FADH2. This FADH2 is inside the succinate dehydrogenase. It's covalently bound to it, and that's why it can send it up through those iron sulfur clusters through heme B into the, I'm trying to think of the right word, inside the membrane to coenzyme Q. Okay, so there are 4 subunits. Yeah, okay. And I apologize once again, I am kind of rushing through this because I don't want to do too much. Okay, so there are 2 things we want to point out. First, we do not send protons across complex 2. Okay, so let's look up here real quick, back at this figure. Complex 1 sends 4 protons. Complex 3, 4 protons. Complex 4, 2 protons. Complex 2, no protons. Okay, this means ultimately that it does not contribute to the proton gradient. We'll talk about that proton gradient and why it's important later, but I just want to draw your attention to that. Okay, so now we've dealt with kind of this part, right? We've gotten electrons to here. We need to talk about coenzyme Q, complex 3, and complex 4. So very briefly, complex 1 and complex 2 are there to bring electrons to ubiquinone to transfer to complex 3. Complex 3 does 2 things. First is it sends protons through. Second is it transfers those electrons to complex 4. Okay, all right. So now let's talk about ubiquinone. And specifically, we're going to be talking about what's called the Q cycle. Okay, so you guys have seen this figure 7 times by now. And in this figure, we see these double arrows. Okay, now we know that oxidation and reduction can't just happen whenever they feel like, and you have to have specific conditions and all that. So let's talk about how we can keep reusing this exact same molecule of ubiquinone and deplete it and regenerate it and deplete it and regenerate it over and over and over again. Okay, the short answer is that we really kind of can't. Okay, anyways, I'm getting a little bit off topic. So first, it's important to kind of pick our starting point. And our starting point is going to be here with UQH2, which is ubiquinol. So it's 2 alcohols. Okay, the first UQH2 is transferred into the P site of complex 3. Okay, so this is all in complex 3 now. We've taken, I guess I should start off by saying C1 and C2 add electrons to Q, which I'll say is UQ, so ubiquinone, and protons are also transferred to make UQH2, which is ubiquinol. This ubiquinol is what we're dealing with. Okay, so then it's moving through the membrane and ultimately it reaches complex 3. Yes, complex 3, sorry. And now while it's in complex 3, it transfers 2 electrons. Okay, so it's oxidized in 2 electron transfers. The first electron transfer is to an iron sulfur cluster. So we're talking about this iron sulfur cluster. So we're in this area right now. I don't know if you guys can see it very well, but this is where we are. Okay, this then transfers it to cytochrome C1 and then to cytochrome C. Okay, so in this process, 2 protons are released. Okay, remember we're doing this to make a proton gradient, so these protons are really important. Okay, so thus far we've transferred 1 electron to cytochrome C. The second electron goes to cytochrome BL, okay, which is just a type of cytochrome B, and then to cytochrome BH, another type of cytochrome B. This then reduces a molecule of UQ to UQ minus. Okay, So now we're going back down to this slide, or we're going to this slide to go to the second half. Okay, so let's talk about what we've just done before we jump into this. First, we've given cytochrome C a single electron. Cool. Second, we've put a second electron. We've had it contribute to creating UQ minus. I don't know what we have to do with UQ minus, but we now have UQ minus. Okay, so we're going to pick up in that exact same place. So we are now right here. All right, and actually, no, I don't want to start here again. I'm going to start at the beginning once more. So now we're taking a second molecule of UQH2, of ubiquinol, in the intermembrane space. So same thing happened. Complex 1 gave us another ubiquinol. We're going to do the same thing with it. Okay, UQH2 in the P site, right? And once again, we have 2 electrons. The first electron, the first electron, we're going to go down with this one. So we're going to go Bl to Bh all the way down to here. And we're going to take this UQ minus, convert it to UQH2 with 2 protons from the intermembrane, from the mitochondrial matrix, excuse me. And now we have a UQH2 that's ready for work. Okay, it's back out into the intermembrane. I'm sorry, not into the membrane space, into the membrane, and it's going to go back and pick up more electrons for us. Okay, the second electron is used, or I guess I should say it follows the same path. So the first electron we talked about last time was an iron sulfur cluster to cytochrome C1 to cytochrome C. So now this is a second electron, and we've released 2 more protons. So we released 2 protons in the first half, 2 protons in the second half for 4 protons. We took in 2 protons to make UQH2, which can then leave, and we've given 2 electrons to cytochrome C. Okay, so the net of the entire Q cycle is this: a single UQH2 plus 2 protons and 2 oxidized cytochrome Cs with a transfer of 2 electrons, kick out 4 protons, reduce 2 cytochrome Cs, and give us UQ. Okay, so I want to point something out that's really weird and also really important. This is 2 separate cytochrome Cs. I drew 2 electrons here to say we've done it twice, but these are 2 separate cytochrome Cs because each cytochrome C gives us a single, so unlike UQ, I guess is the way I want to word this instead. Unlike UQ, cytochrome C can only accept a single electron. Okay, all right. So this is the transmembrane portion of complex 3. It's overwhelming, but interesting fact, this was discovered by a guy at UT Southwestern. Okay, so cytochrome C and UQ differ in a few ways. UQ, first, solubility. So this is hydrophilic. This is hydrophobic. All right, second is this can only accept and donate a single electron at a time. This does 2. Okay, those are the 2 big ones that I want to talk about. Okay, all right, so we talked about electrons being accepted, let's say, by complexes 1 and 2, transferring all the way through those till the end, and then using the Q cycle, ending up in complex 3. Okay, and we're going to pick up from complex 3 in a little bit. But yeah, so that's what we've done so far. We've then, we already have kind of talked about complex 3, haven't we? Because we talked about iron sulfur clusters, cytochromes Bl and Bh, cytochrome C1, then ultimately to cytochrome C. As a reminder, there are 2 cytochrome Cs that both get reduced by single electron transfers and ultimately are going to go to complex 4. And in the process, thus far, we've pushed 8 protons onto the intermembrane space side of this. So we should be okay with everything to the left of that dotted line. Okay, all right, so cytochrome C, like we said, it's soluble mobile electron carrier, reduced by 1 electron from complex 3, which is released to complex 4. Okay, complex 4 is kind of intuitively called cytochrome oxidase because it oxidizes that cytochrome C. Okay, then it uses the electrons released to reduce oxygen to water. Okay, all right, so real quick, oxygen is the terminal electron acceptor, makes this oxidative phosphorylation. Okay, all right. Cytochrome oxidase has, so remember this is complex 4. Okay, has 3 subunits, 1, 2, and 3. Has the redox center that have the redox centers. Then there are 10 others that don't participate in electron transfer. Okay. So there are 2 hemes. We talked about hemoglobin before. Heme is, similar, and 2 copper centers, and 4 electrons are required. Okay. So there are 2 things I'm going to mention here. The first is I'm just going to go through this process real quick. So cytochrome C has an iron that's picking up and donating the electron. Okay. So first iron is going to, first cytochrome C is going to dump its proton, its electron. Keep doing it. The second one is then going to come, so this thing leaves and another one comes in, and then it dumps its second one. So now we're at 2, and for the rest of this, we're going to be dealing with 2. Okay, so this is copper center A. A just because it's the first one, really, right? And then it donates those 2 electrons to cytochrome A. And then cytochrome A donates them to copper B, which is attached to cytochrome A3. And then copper B, finally, after all this time, reduces oxygen to H2O. Okay, so that's the process. Now the second thing, and arguably the more important, not more important, but trickier thing that needs to be emphasized, is 4 protons are pumped for each oxygen released, and yet every figure has shown 2. And this is the reason right here. We tend to deal with 1/2 O2s. We don't like to say 2 O2 or 1 O2 or whatever. We like to say 1/2 O2 gives us 1 water. Okay? And so because we have the O2, we have the proton number with it. Okay? And so all of this that we've talked about should be doubled. And when I say all of this, I mean from the beginning of the electron transport chain till now, should be doubled in order to count for one O2. Okay? So this is another, there we go. This is another drawing of that same thing. And then this is the protein shape. So interestingly, we talked about there being 3 complexes in complex 4, but really the entire electron transport chain could be seen as kind of a super complex. Okay, because it all kind of does the same thing. Yeah. Okay, more images to help you guys conceptualize. All right, so I told you guys the reduction potentials follow this kind of ladder. It goes 1, then we work our way up the reduction potentials. Well, I want to show you guys this in terms of the exact same thing. Okay, so this is generally what's called the downhill path. And it's kind of the opposite because the reduction potentials are flipped. Okay, so reduction potentials work their way up. This is doing the same thing, it's just flipped to the scale. So now up is this way, or positive is this way. Okay, so we start here with NAD plus and NADH, and we can easily transfer to FMN. It takes a little bit of energy to transfer to an iron sulfur cluster, but from there, everything else is down in energy. Okay, and then complex 2 does something very similar. FAD admittedly does take a bit of energy, but everything else is spontaneous. Okay, ubiquinone from both of these, complex 3, all the way down. It follows a generally downhill path, making it pretty much spontaneous. Okay, until obviously, ultimately, it reaches oxygen. Okay, so now this is an interesting discussion. So Mitchell's chemiosmotic hypothesis, and I'm going to read this verbatim, and then we're going to actually kind of break it down to understand what it means. The potential energy associated with the proton gradient across the inner mitochondrial membrane provides the driving force for the ATP, or for the synthesis of ATP. Okay, so blah 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 blah. Okay, this entire thing right here is basically saying the proton gradient we just made. So the potential energy from the proton gradient is basically saying the work that having this gradient can do. So the proton gradient provides the driving force for the synthesis of ATP. So I think this is absolutely brilliant the way that that Mitchell set up his hypothesis and tried to test it and prove it. Okay, I'm not going to spend too much time on this. We haven't gotten to the ATP synthase yet, but this is just kind of a representation of what we're dealing with. So So yeah, so the stoichiometry isn't 100%, but generally we say around 6 protons are transferred out of the matrix, so into the intermembrane space, and about 10 for NADH. So succinate, which I think of as the FADH2, because that's where it's made in the actual Krebs cycle, and then 10 for NADH. So that's per molecule of each. Okay. So the free energy associated with the movement of protons up the gradient has 2 components, the concentration gradient and the electrical potential. In other words, if I have one side, and I'm going to keep this very, very simple, we know that based on concentration, based on diffusion, right, that the definition is it wants to go down its concentration gradient. We're trying to keep things going up their concentration gradient by stuffing more protons in an area that's already got a really high proton gradient. Okay, so that takes energy. Second part is the electrical potential. In the same way as concentration, if one side is really, really positive and the other is really, really negative, the positives should want to flow to the negative. So we're trying to force more protons into an area that's already got a lot of protons. Okay, but anyways, so we've got the proton gradient, which is the electrical interaction. And then we've got the potential difference. Okay, so these are the typical values. Blah, 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 blah. They do a bunch of math. What they get at the end of it is 23.3 kilojoules. In other words, it's non-spontaneous and protons have to be moved uphill. However, by that same exact logic, protons want to move downhill. The process is exergonic for protons to go down their concentration gradient, down their potential gradient, down their charge gradient, whatever we want to call it, because there's a million different names for it. Therefore, we can use that energy. This is potential energy. Okay. And again, these are a lot of things that kind of help us, but I'm not going to spend too much time on them. All right. So now we're finally going to look at the last part of the electron transport chain. The part that actually, the payoff, as I've been calling it, the thing that makes ATP for us, ATP synthase. So ATP synthase is broken into 2 parts, F0 and F1. F0 is kind of in what's it called, transmembrane. Okay, and so it's kind of stuck how it is. It's got A and B and C subunits, cool, and it forms a pore through which protons move. So protons are obviously polar. So if we look at the membrane, this entire area is hydrophobic. How is it going to pass this area? Ignore those lines there. We create a channel. So these pores basically provide the channel for us. Okay, and then the F1 component. So this is a little bit weird. It's got a 3 alpha, or I'm sorry, alpha 3, beta 3, gamma, delta, epsilon structure. So it's got 9 subunits. 3 are the same as each other. 3 are the same as each other, and then there's 3 one-offs. Okay. All right. So another graphical picture, or another picture that helps us. Okay. So let's very briefly actually talk about what we're doing. So first of all, the alpha and beta subunits are these at the top in an alternating pattern, and they get rotated by the gamma and the C subunit. So the gamma subunit rotates, the C subunit rotates. Okay. When they do, the A and B, or I guess I should say, the A and B subunits down here in the F0 part. So there's my mouse. This is the B, and this is the A. These guys stay pretty much still. C and gamma are the ones that rotate. Okay, and you'll notice that gamma, sigma, epsilon are all kind of drawn at once, just kind of by notation. So another important thing to point out is, remember, the periplasm is the intermembrane space. So this is the area that's got a bunch of the protons, right? The protons get transferred in. This is the matrix. Okay, so where is ATP synthesis? It happens in the matrix. Okay, it happens associated with the membrane, but the ATP that's made is made in the matrix. Okay, all right. So these are the C subunits that we've been talking about. A proton that's taken up, and this is really complicated, so we're going to kind of slow down a little bit for this. I know I've been rushing through most of it, but I really do want to slow down because this is really important and I want to emphasize it. So a proton taken up from the intermembrane space through a channel in the A subunit protonates the side chain of an aspartate residue on an alpha helix of the C subunit. Okay, so that's pretty wordy. But basically, the C subunits are alpha helices, right? So this is a C subunit over here, and this one, and this one, and this one, and so on, right? It gets protonated. Makes sense, right? It's a proton that's taken up. All right. Then the C subunits rotate until that proton is lined up with the exit channel. Okay. So it's a little bit weird, but basically, I apologize, there is my mouse. We take a proton, stick it onto here, let it move, and then kick it off. Okay. So think of it almost like if you have two doors that should be right in front of each other, but they're not. One door is in front, you take that door, and then you have to turn right, and then turn and get into the door, to the second door. So what this is doing is it's helping you turn right so that you can guide your way to the second door. Okay, that's it. All right. But that rotation is, well, I mean, that's how it rotates, or that relationship between them is how it rotates. So I've decided to, sorry, you guys can see C3 that I've labeled. I'm going to go ahead and label the other two. C1, C2, and C3 are what I'm going to be calling them. Okay. So this is the alpha subunit. So we talked about it's an aspartate residue on a C subunit that gets protonated. Okay. So you guys here, C1 already has been protonated. C2 and C3 have not. But here shows C3 is now getting protonated. Okay, so we have C1 protonated, C2 protonated, I'm sorry, C3 protonated, C2 is not protonated. All right, well, what happens now? The proton of the C1 subunit, and the 1, 2, and 3 are completely arbitrary, it's really just one of them, swings around. Okay, it swings its way all the way around, kind of pushes its way in. So it kind of shifts everything to the right a little bit, and it kind of kicks off C3. We don't really care about C3 right now. In doing so, it can get really, really close to the alpha subunit and transfer its proton to the serine residue. Okay, why does it transfer it to a serine residue? First, because it can. Second is this is that kind of transfer we were talking about. So it's not the same proton. I really want to point that out. We protonate C3, and then we release a protein from we release a proton from C1, excuse me. All right, so that's the basic process that causes this rotation. So see here is exactly that. This proton will make its way in. Let's say it hits, and I know there's C10 through C15. I just used C1 for convenience, 1, 2, and 3. So let's say it gets this proton protonates this subunit. Well, this entire, I guess, circle, this entire pore, if we want to call it that, is going to rotate its way through, each subunit releasing a proton to that serine residue to kick out, until this proton makes its way all the way back to here, and then it's finally kicked out. Okay, so there are different protons that are protonating the C subunits as are being released. Okay, all right. So it drives the rotation. The rotation of gamma. So this is something that I don't like about the slides as much, is we make it seem like gamma is rotating, but alpha, beta are not. But they induce conformational changes, right? And so that's why I've been kind of shying away from talking about alpha and beta. But the fact is, they induce conformational changes, which then provide the energy to phosphorylate ATP. Okay, so this we're specifically talking about. Excuse me. The alpha beta. The 3 alpha subunits or the 3 gamma subunits or what have you. Okay. So we're going to start off at the top. And before we start off, it's important to know there's the loose, tight, and open subunit. Okay. Now only the tight subunit is active. Okay. The other two are inactive. The open has a very low affinity, and the loose has an affinity, but it kicks off really easily. It's not very consistent. Okay. So the first step, or I guess I should say, this is a cycle. And so in this cycle, the tense subunit starts with an ATP bound. Okay. When the tense subunit has an ATP bound, it then allows ADP to make its way into the loose subunit. Okay. The loose subunit then has a conformational change to make it a new tense subunit. So this loose became tense. And as a result, the originally open subunit became loose, and the originally tense became open. Since it's open, it can kick off its ATP very easily, and that's gone. Okay, because remember, open is inactive and has a low affinity, right? And when ATP is kicked out, we also hydrolyze, and the inorganic phosphate now gives us ATP. So this ADP is converted to ATP, and we can restart. So the next step would be ADP and inorganic phosphate make their way to the new loose subunit. You have conformational changes, and it keeps repeating over and over and over again. And that's how we make ATP. Okay, so these again are all representations to help you guys kind of see what's happening. I'm not going to spend too much time on them because I have about 15 minutes and like 3 more topics. Okay, so let's talk numbers. I told you guys about 10 protons per NADH. And the reason is, so we're ignoring, complex 2 for now. Complex 2 is important, we're going to talk about it next, but complex 2 is not the site NADH enters. Complex 1 is the site NADH enters. And from complex 1, it releases 4 protons, is then transferred to complex 3, where another 4 protons are released, and then transferred to complex 4, where another 2 protons are released. And in the process, we make water. Okay? And then one thing to mention is succinate dehydrogenase, only has 6 because it does not go through complex 1. So complex 2, succinate, I'm going to abbreviate it dehydrogenase. Yeah, DH is fine. Only releases 6 protons because it doesn't go through complex 1. Okay, all right. So now the P to O ratio. So this is weird. So it's the number of ATP molecules made per 2 electrons that enter the electron transport chain. Okay, so first, let's start off by saying one complete, I'm going to start off with this bold term, and then we're going to work our way back. So the human body, we have 12 C subunits of ATP synthase. Now, we talked about the 3 subunits alternate between that tense, loose, open, tense, loose, open, and so on. And so if we think of it as one complete turn of the enzyme, means this thing has gone from tense to loose, then loose to open, then open to tense. But the other 2 subunits have done the same. And so each subunit makes 1 ATP and then goes back to its original state. The loose, open, tense, and so on has made 1 ATP. And then the open, tense, loose has also made 1 ATP. Therefore, per 1 complete turn of ATP synthase, we generate 3 molecules of ATP. Because we have 12 subunits, and we talked about how the subunits basically rotate all the way around to release a single proton, each subunit has to get protonated to turn the enzyme 1 complete cycle. Therefore, the number of subunits equals the number of protons for 1 complete turn. 1 turn equals 3 molecules ATP. Okay, so now let's come back to this. So where am I? Sorry, I got lost. The number of ATP molecules made per 2 electrons. Okay, well, we have the number of ATP molecules is 3 ATP per 12 proton. Helps us, doesn't help us too much. But when we look at the second bullet point, now we're getting somewhere. Na deoxidation is 10 protons per 2 electrons. 10 protons per 2 electrons. So what can we do to get ATP per 2 electrons is multiply these 2. So 10 times 3 over 12 times 2 is 30 over 24. Right? Yes, okay, I did that math right. Oh, no, I apologize. It is not over 24. The reason it's not over 24 is because we have to look at 2 electrons. I always miss this, okay? So think of 2 electrons as a unit. There's 1 2 electron unit, 1 2 electron unit, so it's 1 times 12. Okay, this is something that I've messed up quite a few times, and that's why I was really paying attention for it this time. So 30 over 12 is 2.5. Okay, in other words, where are we? I know it says 2, but there it is. Okay, so if 1 ATP is made per 4 protons, so what they did is they said 12 divided by 3, so H plus to ATP is 4. Therefore, 1 ATP per 4 protons, which is basically the same as that 312, this 3 over 12 number, just reduced. And then we said 10 protons, so we can make 2.5 ATP per 2 electrons. In other words, 2.5 ATP per 1 NADH. Okay. Now, this is a really, really important point. In mitochondria, so eukaryotes, we use up 1 proton to export each ATP. So I told you all last week, I think it was last week. Yeah, it was last week. No, not last week. I told you all earlier today. What am I saying? I told you all earlier today, the ATP is made in the matrix. Well, it's great, but sometimes we need it outside the matrix, and therefore it takes an extra proton. And so we think of our actual P to O ratio as 2. Okay, yes, it is 2.5, but it's 2. Okay, so this is a generic formula that I think is really, really important to memorize. Okay, so P over O equals, so 10 is number of protons. Number of protons. Okay, we're going to divide that by protons per ATP, ATP plus 1. Okay, so protons per ATP is this 4, which they got from 12 protons per 3 ATP, and then they added 1, adding 1 because the 1 for the ATP transport. Okay, so this question right here is absolutely 150% going to be on your exam. I can tell you right now, I'm almost certain of it. Okay, so the reason I'm telling you to memorize this is because this is the easiest way to do it, and this plus 1 is only in eukaryotes. Okay, so we're going to do 2 practice questions real quick over to this side. So I'm going to tell you, I've discovered a species that has 16 C subunits. Okay, and it's a bacteria. No, we're going to say no, it's not a bacteria. This is a eukaryote. So I add 1. Okay, so I need a moment to actually calculate this because I totally did not make this question beforehand. I just made this real quick. 1. Okay. The number I get out is about 1.6. And yes, there's decimal points. I'm going to round to 1.6 for now. So this is saying per 1 molecule of ATP, I'm sorry, I make 1.6 molecules of ATP per 2 electrons into the electron transport chain. Now, what if the same thing were bacteria? Same exact situation, but it's bacterial. Well, we're going to set up the formula the same, 10 over 16 over 3. We are not going to add the 1 because we no longer need that 1 for electron transport. So therefore, this number jumps up, well, it doesn't really jump up, but it jumps up to almost 1.9. It's 1.88. Okay, so now it's a little bit more efficient in bacteria than in eukaryotes. Okay, and that's what it says right here. In bacteria, ATP is not exported, so P to O ratios are higher. Okay, all right. So I told you guys about Mitchell's hypothesis. The chemiosmotic hypothesis basically said, it basically said we think that the potential, that the powering of ATP synthesis comes from the proton gradient. Okay. Well, the Racker and Stachinius experiment proved Mitchell's hypothesis, this chemiosmotic hypothesis. Okay. So bacteria rhodopsin. Rhodopsin, for those of you who don't know, is in your eyes. Okay. Bacteria rhodopsin is slightly different because it's a light-driven proton pump. So when it's hit with light, it can push protons up their up their gradient, in this case, into the membrane that they've created. Okay, so they made these little lipid vesicles, which is just a bilayer basically, and they've said we're going to put bacterial rhodopsin here, and we're going to use that to push protons in. Okay, and then we're going to also attach ATP synthase to this vesicle, and we're going to see what happens. Okay, and they found ATP does get created. Okay. I think there was another slide about, okay, no, there wasn't. Okay, that's fine. So what if they'd found that ATP wasn't created? Well, then they could say the proton gradient isn't what's causing ATP synthase to spin. Okay, all right. So now we're going to talk about what are called uncouplers. Okay, so I have seen a few of these on exam. They're fair game. But basically, the whole point of the electron transport chain is to create the proton gradient. Then the proton gradient spins ATP synthase. Okay, so because of that, there are certain inhibitors that can target specific points and kind of ruin that. Okay, so the points that they can specifically target are uncouplers just naturally increase how many protons can transfer. So generally speaking, we have a few pores or proton channels where H plus can go back through. Now, ultimately, these guys are pushing through 4 protons at a time, and so it's a little bit faster, and they're what are keeping up the proton gradient. However, uncouplers speed this process up significantly. So they increase the proton conductance of the membrane, and so they cause a short circuit. They cause the proton gradient to collapse. Okay. We could also target complex 1 using these 3 drugs, any of these 3 drugs, I should say. We could also target complex 4 or cytochrome C oxidase using any of these 4 drugs, or 3 drugs, excuse me. Or, once again, we could target ATP synthase with oligomycin. Okay, so all of these different places are places where, if we want to think of it as this, we could effectively say we're inhibiting the electron transport chain in ATP synthesis. Okay. All right. So DNP for you guys is dinitrophenol. It's right here. So it's an uncoupler. Okay. Dinitrophenol is kind of hydrophobic. It's kind of hydrophilic. Okay. So even when it's charged, it's hydrophobic enough because it's got that charge delocalization of an aromatic ring, because it can resonate so well. It's kind of pseudo hydrophobic, if you want to think of it like that. So what it does is DNP minus travels across the membrane. It picks up a proton and it travels back across the membrane. So in doing so, what it's effectively doing is it's stealing these protons from in this case, the intermembrane space or the periplasm, and bringing them back to this side. So if there were, let's say, 10 protons here and 1,000 here, well, this can start bringing this number down and down and down, and this number up. And remember, the importance of the proton gradient is that there's a big difference between the two. So if you bring 10 up to 200 and this down to 300, you've basically eliminated the proton gradient, right? It takes one more step and it's done. And so that's what some of these uncouplers can do. Okay. So I told you guys this is just like a battery. Well, here's your example, or here's your proof, I guess. Your professors agree with me. So what we're doing is we're basically saying electrons go to oxygen to create water. Okay. That in turn releases protons, which can then spin ATP synthase to do something. And then they go back around and we repeat the process, right? The exact same thing happens in an electric circuit. Your battery, this is a symbol for a battery, releases electrons, electricity, to do something, in this case, light a light bulb, and go back around. So the electron transport chain provides the potential difference the same way a battery would. Protons provide the current the way electrons would, and ATP synthase is the work that's done as opposed to the lighting of this light bulb. Okay. So an uncoupler cuts this process in half. Current flows and everything, but we get rid of the part where we actually do work. And so therefore energy is just dissipated as heat. We've gotten rid of the actual work. So that's why it's short circuit. Okay. All right. So yeah, so this is another picture of that. All right. So natural uncouplers are good, right? They can provide a source of heat for us. We do need natural uncouplers. It's just the unnatural uncouplers that target the electron transport chain are bad for us. Okay. All right, now this is something interesting your professors like to include because there's a lot of dieting and a lot of talk about fat and what kinds of fat and tissue we want and all that. I think it's interesting. I don't remember if it's ever been asked about. I think maybe once or twice they might have asked a question about it. So just very briefly, excuse me. So basically, fat is stored in white adipose tissue. Okay. Brown adipose tissue can be activated to burn fats and sugars. Okay. So and I'm just going to highlight this last part at the end. So BAT can be activated in response to various stimuli, including exposure to cold, to burn fat and sugars. So it's good for us. Now the process seems to be more prominent in the young and lean than in the cold and obese, and in women rather than in men. Or, I'm sorry, did I say cold? I meant old and obese. So in other words, this factor, this presence of brown adipose tissue, this activation of brown adipose tissue, can contribute to weight loss, or it can contribute to weight in general. Because if, let's say, my brown adipose tissue is less prominent, then it's not responding to various stimuli, and so I'm burning less fat and sugar than someone else might. Okay, and so this could be a factor affecting weight and obesity and all that. It's just a really interesting thing that a lot of professors like to include because it is quite interesting how this stuff works. Okay, and that's actually it. And I think I've gone about 2, maybe 3 minutes over. So thank you all for your time and for really this entire semester. I've had a lot of fun teaching these reviews. And good luck on your final. If you ever need anything, please drop by tutoring this week or yeah, let me know anytime.